Okay, so we will now move to the next talk. I see Michael waiting there. So Michael is currently at the Cold Spring Harbor Laboratory. He got his degree at the University of Maryland, and he sort of shocked the world by producing some of the most sophisticated uses of MapReduce for key biology applications, and I assume he'll tell us about that work and what the futures are. Thank you very much.
Okay, yeah, so if there's any problems with audio, please just let me know, otherwise I'll continue on. Okay, so back to the figure. So at the top, we have a little cartoon. We have our DNA. So for the human genome, that's about 3 billion base pairs. Pieces of it will get transcribed into these little RNA transcripts, exported outside of the cell nucleus. The ribosome then knows how to read those off into chains of amino acids, and those amino acids form proteins. So what's remarkable about this is that your genome and your environment basically totally define who you are. So everything from your physical form as being a human with 5 fingers and 5 toes, to your hair, eye color, skin color, all those things. But more significantly, susceptibility to diseases, responses to drugs, all have a very strong genetic component, as does personality, social disorders, many different characteristics are all driven by your genome. As a result, there's a tremendous interest to sequence genomes and to sequence the RNA and to really study these cellular activities. So they're just really fundamental and important questions like, what is your genome sequence? How does your genome compare to my genome? Where are the genes in your genome? How active are they? How does the activity change under development, and so forth? Really, the list of questions goes on and on and on and on and on related to all these cellular processes. So the very earliest methods for sequencing DNA, and the methods are quite similar for RNA, go back to the late 70s. And this was a totally manual method for sequencing DNA, where your sample was tagged with special radioactive nucleotides that you could then shine an X-ray on and be able to read off the sequence of nucleotides. It was very, very manually intensive, very, very low throughput, but for the first time we were able to read off the complete genome for an organism, this little tiny virus that was about 5,000 base pairs long in 1977. In the 80s and 90s, we saw the development of automated sequencing machines, and that led to discoveries such as in 1995, the first free-living organism was sequenced. That was this little microbe called Haemophilus influenzae that was about 1.8 million base pairs long. The most famous example of this first generation of automated sequencers was the publication of the human genome in 2001 that really brought in it was really a landmark discovery in that for the first time we had all of the genetic content of a few human individuals. That discovery was primarily enabled by improvements to biotechnology and compute technology. In particular, there was a famous investigator, Craig Ventnor, that went on the record saying that the automation was so revolutionary that it could decode in a single day the same amount of genetic material that most other DNA labs could produce in a year. So it was one of these orders of magnitude improvements to the biotechnology gave rise to these first early genomes that were sequenced. In the 2000s, we saw the rise of so-called second-generation sequencing, and we saw instruments from companies such as 454, Illumina, and Life Technologies really enabling really massive amounts of sequencing to be performed. So rather than thousands or hundreds of thousands of base pairs per day, now we're talking about on the order of billions and billions of base pairs per day. So we can just with tremendous throughput, we can sequence all these genomes and collect huge amounts of biological data very, very efficiently. The innovations continue, and we're now entering what's sometimes called the third generation of sequencing, with new instruments that have new capabilities and even a very portable footprint. So in particular, in February, there was an announcement from a company, Oxford Nanopore, they were planning to bring to market a new device about the size of a thumb drive. So there's a picture of the instrument in someone's hand. that would be able to sequence the human genomes, they say, in about 15 minutes and for about $1,000. So this is going to dramatically change the nature of biology and sequencing, because suddenly we can have this very portable device that can inexplicably make measurements and sense the biological environment around us. Just to give you a little sneak peek at how some of the sequencing works, so it's in some sense a very, very fancy camera, if you will. So we're going to take our molecules of DNA, we're going to attach them to just a glass slide, just to lay them out onto a field. We're going to grow up many copies of the DNA, and at the end of the DNA, we're going to attach special fluorescent nucleotides that are glow a certain color. So we're going to, for all these molecules on this glass slide, we're going to attach a fluorescence to the very end of all of them, take a picture of it. So those will glow one set of colors. Then we're going to attach fluorescent nucleotides to the second base pair, take a picture of it. That'll glow another set of colors. And then the third base pair, that'll glow another color. And as a result, these instruments, in some sense, will expose the stack of images and the colors of those images reveal the sequence of all the different molecules. So on the right, we have a little cartoon with a couple of different images, with a couple of different dots highlighted. So one goes green, 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 all the way through the stack. So that's like T, 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 T. Whereas the other one goes green, that's a T, blue, that's a G, red, that's an A, T, that's another, green, that's another T, and so forth. So already there's this huge computational problem to be able to do the image analysis, to be able to take this stack of raw images and then translate that into DNA nucleotides. This process, though, is only reliable, with this chemistry, is only reliable for about 100 consecutive nucleotides in a row, although over really massive throughput. So we're going to get on the order of about 1 billion of these 100 base pair, 100 nucleotide segments in about a 9 or 10 day run. So these instruments are being applied to many, many different studies from investigators around the world. So here I have the logos from a few different projects. So the first logo is the logo from the 1,000 Genomes projects, using massive amounts of sequencing to study the diversity in healthy human beings from around the world. The next logo is from the Cancer Genome Atlas project that is using deep sequencing to study how cancer mutates through genomes and then also develop drug therapies in response to those mutations. There's a project called the Genome 10K project that aims to sequence 10,000 vertebrate animals, and that'll give us enormous power to look back in evolutionary time to see how different genomes and how different species came to arise. There's a project called ENCODE that is using deep sequencing to measure all the biologically active regions of your genome. And then there's another project called the Human Microbiome Project that is using deep sequencing to study all the organisms that live in our body and inside our body. What's interesting for all of these different projects is that the raw sensor data from these instruments never give us the answer that we're looking for. In every single case of these projects and countless other projects, there needs to be very large-scale computation to be able to take those short sensor data and then turn them into intelligible results. So one of the most fundamental computations available is something called short read mapping. So the idea is we have our long reference genome assembled for, say, the human genome. Then we can apply various string matching algorithms to those short reads to align each of those short reads to the long reference genome to figure out where it occurs. And then here's an example where the reference genome is an A, but all of the measurements that we've just made are T's. So that tells us that, oh, this person was mutated at this position from A to T in their genome. Another fundamental computation very closely related is some sort of what I'm going to call a differential analysis. So if you collect the biologically active parts of your genome, your RNA, your gene regions, under one condition, you might see a pattern in green. If you collect the RNA under a different condition, you might see the pattern in red. So that can tell you how your body is reacting to different stresses. So this could be, the temperature is different, the nutrients are different, you're being attacked by a virus, you're being attacked by some other stress in the environment, and then we can study how your body, how your cells change in response to that. Another very fundamental computation is called Denovo Assembly. So taking all of those short reads, comparing them to each other to build up some sort of graph structure, and then analyzing that graph structure to be able to reconstruct the original genomes. So it's that computation that allows us to take, say, 100 base pair reads and then stitch them together, much like assembling a jigsaw puzzle into a complete genome that's billions of base pairs long. And then very generally, there's a lot of interest to take genomes, to study their phylogeny, to see how they relate to each other, and then also to ask questions like, if this genome was changed in this way, what would be the change in response to their traits? So, you know, if it's a plant, maybe we can modify the genome a little bit so that it grows taller, or maybe the fruits are sweeter or bigger, or it's more disease resistant and so forth. And all that is done through computational modeling to try to determine, you know, how the organism development will change in response to the change in the genome.